will process those inputs through the digestive tract, through the liver, through the kidneys, through the ordinary machinery of metabolism. Fine, adequate, functional, but when a photon of ultraviolet light strikes your skin, something happens that no pill can replicate. Not because the chemistry is exotic, but because the delivery system is fundamentally different. The photon does not pass through your stomach. It does not wait in line behind your breakfast. It arrives at the precise molecular target it was always going to hit, at the speed of light, with exactly the energy required to do one specific thing, and that one thing sets off a chain of events so extensive, so deeply wired into your physiology, that calling sunlight a vitamin source is like calling the ocean a glass of water. Let me tell you what actually happens. Not the simplified version. The real version, the version that involves quantum mechanics, evolutionary biology, orbital mechanics, and the reason your sleep fell apart 3 years ago. There is a molecule in your skin called 7-dehydrocholesterol. You made it yourself. Your cells synthesized it from cholesterol and transported it to the outer layers of your epidermis, the stratum basale and stratum spinosum, where it sits embedded in cell membranes like a satellite dish pointed at the sky. This molecule has a hexagonal ring structure, 6 carbon atoms bonded in a ring, and one of those bonds, the bond between carbon 9 and carbon 10, has a very specific energy threshold. It takes exactly 4.4 electron volts to break it. Not 4.3, not 4.5. The bond has a resonance frequency, and a UVB photon between 290 and 315 nanometers carries precisely that energy. When the photon arrives, the bond absorbs it and snaps open. The ring unfolds. The molecule changes its three-dimensional shape in a fraction of a second. It is no longer 7-dehydrocholesterol. It is now pre-vitamin D3, a different molecule with a different geometry and a completely different biological destiny. Now here is the part that should make you pause. Your body put that molecule there on purpose. It synthesized it, transported it, and positioned it in the exact tissue layer where UVB photons penetrate. Your skin is not passively receiving sunlight. It is actively waiting for it. The molecule is an antenna. The photon is the signal. And the system was designed, by 400 million years of vertebrate evolution, to operate this way. Fish make vitamin D in their scales. Reptiles make it in their skin. Birds make it in their feathers and then ingest it during preening. Every vertebrate on Earth uses ultraviolet light to drive this reaction. You are not an exception. You are the latest iteration of a system that predates the existence of bones themselves. After the ring opens, your body temperature does the rest. 37 degrees Celsius, the heat of your own blood, slowly rearranges the pre-vitamin D3 molecule over the next 24 to 48 hours through thermal isomerization. No enzyme required. No cofactor needed. Just the ambient warmth of being alive converts it to cholecalciferol, vitamin D3. From there, your liver hydroxylates it once, producing calcidiol, the form your doctor measures in blood tests. Your kidneys hydroxylate it again, producing calcitriol, the active hormone. And calcitriol does something that no other vitamin does. It enters your cell nucleus and binds directly to DNA. It is a transcription factor. It sits on your genome and tells genes to turn on or stay silent. Over 200 genes respond to its presence. Genes governing calcium transport, phosphorus regulation, immune cell activation, cell proliferation, apoptosis, insulin secretion, neuromuscular function. 200 genes. From one photon. From one broken bond. From 15 minutes outside. A supplement pill skips the first step. It gives you cholecalciferol directly, bypasses the skin entirely, and enters through the gut. And the gut absorbs it with variable efficiency depending on fat content of the meal, bile acid availability, intestinal health, and a dozen other factors. The bioavailability is inconsistent. The dose is fixed, and there is no feedback mechanism. Your skin has one. When enough pre-vitamin D3 accumulates in the epidermis, further UV exposure converts the excess into inert photoproducts, lumisterol and tachysterol, which have no hormonal activity. The system self-regulates. You cannot overdose on sun-produced vitamin D because your skin has a ceiling built in. The pill has no ceiling. The pill relies on you reading the label correctly and your doctor monitoring your blood levels periodically. The difference between the photon pathway and the oral pathway is not just route of administration. It is the difference between a self-regulating biological system and an unregulated chemical input. Michael Holick at Boston University spent decades quantifying this. 15 minutes of midday summer sun on arms and legs produces between 10,000 and 20,000 international units of vitamin D3. A standard supplement contains 400 to 2,000 units. Your skin, given a quarter hour of sunlight, manufactures 5 to 50 times what a pill provides. And it does so with perfect dose control, stopping production automatically when stores are sufficient. Holick published this data repeatedly. He was removed from the dermatology department at Boston University for advocating moderate sun exposure. The institutional position was total avoidance. The photochemistry supported a more nuanced conclusion. The politics did not care about the photochemistry. But vitamin D is only the beginning of what sunlight does. And this is where the story becomes something a pill cannot touch at any dose. Behind your retina, layered beneath the rods and cones that give you color vision and edge detection and the ability to read these words, there is a population of cells that have nothing to do with seeing. They were discovered in 2002 by David Burson at Brown University, and they are called intrinsically photosensitive retinal ganglion cells. They contain a pigment called melanopsin, which is maximally sensitive to light at 480 nanometers, deep blue, the color of a clear morning sky. These cells do not send signals to your visual cortex. You do not perceive their activation as sight. They send signals along a dedicated neural tract, the retinohypothalamic tract, directly to a cluster of 20,000 neurons in your hypothalamus called the suprachiasmatic nucleus. This cluster is your master clock. It times everything. When cortisol surges in the morning to wake you, when body temperature peaks in the afternoon, when melatonin rises at night to initiate sleep, when growth hormone pulses during deep sleep, when hunger signals fire, when attention sharpens, every circadian rhythm in your body, and there are hundreds of them, takes its timing cue from this cluster. And this cluster takes its timing cue from one input only: light entering your eyes through melanopsin. The critical number is lux. Indoor lighting, the light in your kitchen, your living room, your doctor's office, produces between 100 and 500 lux. The melanopsin system requires approximately 1,000 to 2,000 lux for robust circadian entrainment. Outdoor shade on a cloudy day produces 10,000 to 25,000 lux. Direct sunlight produces 50,000 to 100,000. The gap between indoors and outdoors is not 10 or 20%. It is 1 to 2 orders of magnitude. If you spend your entire day inside, your circadian clock is receiving 1/50th of the signal it evolved to expect. It is like trying to tune a radio when the transmitter is barely above the noise floor. The signal is there, technically, but the clock cannot lock onto it cleanly. Without a strong morning light signal, the human circadian clock drifts. Its intrinsic period is approximately 24.2 hours, measured in isolation experiments where subjects lived in bunkers without time cues. That extra 12 minutes per day sounds trivial. Over a week, it shifts your sleep-wake cycle by nearly 90 minutes. Over a month, you are living on a completely different schedule than the sun demands. Morning light resets the clock to exactly 24 hours every single day. It is the synchronization pulse. Without it, the system free runs, and free running in a world that operates on solar time means chronic misalignment between when your body wants to sleep and when your life allows it. If you are over 65 and your sleep has deteriorated, if you wake at 3 in the morning and lie there for hours, if you feel exhausted by early evening, if you never feel sharp during the day, the most likely physical explanation is that your circadian clock has lost amplitude. The peaks have flattened. The cortisol morning surge is shallower. The melatonin evening rise is weaker. The system has lost contrast. And the reason it has lost contrast is almost certainly insufficient light input. You are indoors too much. The melanopsin cells are not firing hard enough to maintain the rhythm. There is an additional insult that aging delivers specifically to this system. The lens of your eye yellows progressively over decades. Chromophores accumulate in the lens proteins, absorbing short wavelength light preferentially. By 65, your lens transmits significantly less blue light than it did at 25. The exact wavelength melanopsin needs, 480 nanometers, is precisely the range your aging lens filters most aggressively. So the signal is weakened at both ends. Less time outdoors means fewer photons arriving at the eye, and the yellowing lens means fewer of those photons reach the melanopsin cells even when you are outside. The circadian system is being starved from two directions simultaneously. The solution is not complicated. It is more time in brighter light to compensate for the reduced transmission, but it requires being outside, not next to a window, not in a bright room, outside, where the lux levels are high enough to overcome the filtering. Sachin Panda at the Salk Institute and others have documented that increased morning bright light exposure in older adults improves sleep efficiency, reduces nighttime awakenings, consolidates the sleep-wake rhythm, and increases daytime alertness. The intervention is not pharmacological, it is photonic. No sleeping pill resets your suprachiasmatic nucleus. No drug enters your retina and recalibrates the clock. Only photons do that, only light through the eyes at the right time of day. Now the melanopsin cells connect to something else that matters enormously for how you feel during the day. They project to the Raphi nuclei in the brainstem, where serotonin is manufactured. Gregory Lambert and colleagues, publishing in The Lancet in 2002, measured serotonin turnover directly by sampling the internal jugular vein, the vein that drains the brain, in living human subjects. They correlated serotonin production with the amount of bright light exposure each subject received that day. The relationship was linear and direct. More photons through the retina meant more serotonin synthesized in the brain, not a subtle correlation buried in noise. A clear, dose-dependent relationship between light input and the brain's primary mood-regulating neurotransmitter. This is the molecule that SSRIs target. Selective serotonin reuptake inhibitors work by preventing the reabsorption of serotonin from the synaptic cleft, keeping whatever serotonin is present active for longer. Sunlight increases the production, the drugs slow the removal. Both raise effective serotonin levels, but through completely different mechanisms. And serotonin is also the precursor to melatonin. Your pineal gland converts serotonin to melatonin after dark. Higher daytime serotonin means more raw material available for nighttime melatonin synthesis. The entire light-mood-sleep axis is one continuous biochemical pipeline. Sunlight enters the retina, drives serotonin production during the day, and the accumulated serotonin converts to melatonin at night. Cut the light input and you cut both ends. Lower mood during the day. Worse sleep at night. Less energy the next morning, less motivation to go outside, less light input. The spiral feeds itself downward. Morning sunlight breaks it at the source. There is a third mechanism operating through your skin that has nothing to do with vitamin D and nothing to do with your eyes, and it explains something about blood pressure that most cardiologists have never considered. Richard Weller, a dermatologist at the University of Edinburgh, discovered that your skin stores nitric oxide in chemical reservoirs, nitrite and S-nitrosothiol compounds distributed throughout the epidermis and dermis. These reservoirs are substantial. They are loaded with vasodilator waiting to be released. The release signal is UVA radiation, the longer wavelength ultraviolet that penetrates deeper into skin than UVB. When UVA photons hit these reservoirs, they break the bonds holding the nitric oxide in storage. The gas diffuses into your bloodstream, reaches the smooth muscle cells surrounding your arteries, activates guanylate cyclase, produces cyclic GMP, and the muscle relaxes. Your arteries dilate, your blood pressure drops. Measurably, reproducibly, through a mechanism that no oral medication targets. Weller controlled for temperature meticulously. He used UV lamps at constant ambient temperature to prove the effect was photochemical, not thermal. He blocked UVB to prove it was independent of vitamin D. The blood pressure reduction was real and operated through a completely novel pathway. He then made an argument that challenged his entire specialty. Cardiovascular disease kills roughly 80 times more people per year than melanoma. If moderate sun exposure reduces cardiovascular mortality even modestly through nitric oxide release, the net effect on all-cause mortality favors going outside, not staying in. The dermatological establishment did not welcome this argument. But the data was published in the Journal of Investigative Dermatology. The nitric oxide measurements were reproducible, and the cardiovascular mortality statistics came from national registries. The physics was not a matter of opinion. Now I need to be honest about the other side of this, because the same photons that do all of these beneficial things also damage DNA. UVB radiation creates thymine dimers, covalent bonds between adjacent thymine bases on the same DNA strand. Your cells have nucleotide excision repair enzymes that detect these lesions, cut out the damaged segment, and rebuild it from the template strand. The system handles thousands of repairs per day per cell, but it is not perfect, and its capacity declines with age. Accumulated unrepaired damage over decades increases mutation risk, and some mutations lead to skin cancer. This is real. It is not trivial. And it means the dose matters enormously. The convergence point, where benefits plateau and damage continues accumulating, is well characterized. 10 to 20 minutes of unprotected midday sun on arms, face, and legs is sufficient for near maximal vitamin D synthesis, significant nitric oxide release, robust circadian signaling through the eyes, and meaningful serotonin stimulation while remaining within the skin's daily repair capacity. Beyond that window, protection makes sense. Within it, the factory runs at full capacity and the maintenance crew keeps pace with the damage. The physics is simultaneous. Benefit and risk operate on the same photons at the same time. The dose separates them. There is one final connection that ties everything together, and it runs through your bones. Calcitriol, the hormone that started as a broken bond in your skin, controls calcium absorption in your intestines. Without adequate calcitriol, your gut absorbs only 10 to 15% of dietary calcium. With adequate calcitriol, absorption rises to 30 to 40%. That difference determines whether your skeleton receives enough calcium to maintain its architecture or whether your body, needing calcium for nerve conduction and muscle contraction and blood clotting, begins withdrawing it from the only large calcium reservoir available, which is your bones. Osteoporosis is, in part, a disease of insufficient photons. The chain runs from sunlight to skin to liver to kidney to gut to bone. Break any link and the skeleton pays. But calcitriol also acts on muscle. Vitamin D receptors exist in skeletal muscle tissue, and calcitriol binding activates genes involved in muscle protein synthesis and neuromuscular coordination. Deficiency correlates with weakness, impaired balance, increased postural sway. Multiple trials show that correcting vitamin D deficiency in older adults reduces fall risk by approximately 20%. The photon that strengthens the bone also strengthens the muscle that prevents the fall that would break it. Both ends of the catastrophe are addressed by the same upstream input, and calcitriol modulates innate immunity. It activates antimicrobial peptides called cathelicidins that macrophages deploy against bacteria and viruses. Low vitamin D means low cathelicidin means reduced pathogen killing. The winter respiratory infection season peaks precisely when vitamin D levels reach their annual minimum at northern latitudes, the months when UVB cannot penetrate the atmosphere at a steep enough angle to drive the skin reaction. Before antibiotics existed, tuberculosis patients were placed on sun-facing balconies. Niels Finsen won the Nobel Prize in 1903 for treating lupus vulgaris with concentrated light. The mechanism underneath, cathelicidin activation through vitamin D, would not be understood for another century. But the clinical observation was correct. Sunlight helped the body fight infection. The physics just took 100 years to explain what the doctors could already see. So step back and look at what 15 minutes of morning sunlight actually does. A photon breaks a bond in your skin and initiates a hormonal cascade that controls 200 genes, builds bone, activates immune cells, strengthens muscle. Melanopsin cells in your retina detect the light and reset your master clock to the rotation of the Earth, consolidating your sleep and sharpening your daytime alertness. Serotonin production increases in your brainstem, lifting mood and providing raw material for nighttime melatonin. UVA photons release stored nitric oxide from your skin, dilating arteries and lowering blood pressure for hours, all from stepping outside, all absent when you stay in. No pill does all of this simultaneously. No supplement resets your circadian clock. No capsule releases nitric oxide from your dermal reservoirs. No tablet enters your retina and fires your melanopsin cells. These are parallel systems, evolved over hundreds of millions of years, all calibrated to the same input: the broadband electromagnetic radiation of a star 93 million miles away, arriving at your skin and eyes at the speed of light, carrying exactly the energies your molecules need to change shape and start cascades. Your body does not crave sunlight the way you crave food. There is no conscious hunger for photons, but the deficit accumulates silently. Sleep degrades, mood flattens, blood pressure creeps up, bones thin, muscles weaken, balance falters, immunity drops, each system declining independently, each treated with a separate medication, each addressed as if it were an isolated problem. But they share a common upstream cause. The raw material they all need is outside. It has been outside every morning since you were born. And the body that was built to receive it, the body that positioned molecules in your skin specifically to catch it, the body that wired dedicated neural pathways from your retina to your hypothalamus specifically to detect it, that body is still waiting, still ready, still equipped with every mechanism it needs to use what the sun provides. The machinery has not broken down. It has been idling, waiting for the input it was designed to run on. 15 minutes tomorrow morning. Face to the sky, arms uncovered, eyes open to the bright world. The photon that left the sun 8 minutes ago is arriving now. Your skin is ready, your retina is ready, your clock is ready, your arteries are ready. Everything downstream, the bones, the muscles, the immune cells, the serotonin, the melatonin, the 200 genes waiting for their transcription factor, all of it is ready. It has always been ready. The only missing variable is you. Outside, where the light is,